is Dr. Ripudaman Singh. He's the Chief Technology Officer at Arcedi Biotech, which is a Danish biotech company who developed a technology for non-invasive prenatal diagnosis on rare fetal cells isolated from the maternal blood.
Thank you very much, and I would like to thank the organizers for inviting me here to speak about our project. So I'm also going to talk about NIPD, non-invasive prenatal diagnosis, but from another perspective, from the fetal cells and maternal blood. What we have done in RCD Biotech is that we have developed a technology by which we can enrich rare fetal cells from the blood of the pregnant woman, and then we can use these rare cells for prenatal diagnosis, something that has been coined as cell-based non-invasive prenatal diagnosis. So you see here similarities with other non-invasive prenatal testing. Well, I don't have many disclosures apart from the fact that I'm the Chief Technology Officer at RCD Biotech, and I'm also one of the core inventors of the technology. So this is the pictorial representation of the RCD goal. So what we would like to achieve is that we would like to ultimately replace the invasive methods, the CBS and amniocentesis, with a simple blood draw from the maternal blood, because we know that there are fetal cells that are circulating in maternal blood, so we'd like to use these fetal cells for prenatal diagnosis. And the rationale behind this goal is that we would like to achieve high fetal genome coverage and purity, and at the same time, when we are sourcing the fetal genome, we would like to have little risk or stress to the pregnancy. And this is just to give you an overview of our technology. So what we are doing, we get blood samples from pregnant women who are coming for ultrasound scan at the end of the first trimester. So they are between the gestational ages of 10 to 13. So we are processing the blood. So what is basically happening is that we are lysing the RBCs. What you are left with is basically white blood cells, and there are some fetal cells among them. Then we have proprietary antibodies. We use the magnetic cell sorting from Miltony. So we conjugate the beads, the antibodies with the magnetic bead, run it through the magnetic column. So what you have in the top is the fetal cells, which are enriched. All the negative cells, the maternal cells, go in the flow through. Then we have another set of specific fetal cell marker that stains the fetal cells. The enriched and stained fetal cell fraction is then smeared on the slides. Then we use a fluorescence microscope, which has an integrated picker. I'll come back to that. We scan for the fetal cells. We positively identify them based on the staining morphology. We pick them individually, perform whole genome amplification, and subsequent genetic analysis using CMA currently. Just to give you a brief historic perspective of the fetal cells in maternal blood, it's been known for years that there are some cells that go from the fetus to the maternal circulation, and this process starts as early as the first trimester. So it was quite obvious that when people were looking for alternative technologies for non-invasive prenatal diagnosis or invasive prenatal diagnosis, they started looking for fetal cells. And it would make sense because it would mitigate the risk which was associated with the invasive methods, but it would also make the prenatal diagnosis simple and cost-effective. However, it was not very easy to source these fetal cells from maternal blood for many reasons, and some of the challenges are listed here. First is that there was lack of knowledge of the type of the fetal cells that circulate in maternal blood, and connected to that was that there were not many markers that one could use to enrich these cells from the maternal blood. And on top of that, everything was made very complicated by the fact that these cells were very rare. So some of the numbers put down these numbers to, calculations put these numbers to one fetal cell among 1 million maternal cells. So you can imagine the challenges that there were in enriching these rare cells. And this is another way of looking at it. So these are the number of publications plotted against the year, looking for fetal cells in maternal blood. So it's an old field, you can imagine. So the first study that was published looking for fetal cells in the circulation was in 1959, but it was not until 1969 that the potential of these cells for prenatal diagnosis was proposed. But then researchers had to wait for another 10 years to develop a protocol to enrich these cells from maternal blood. And then the so-called golden era of fetal cell research, where a lot of data was being churned, was between 1993 to 2003. But coming back to the challenges of enriching these cells from maternal blood, so you can imagine how much interest this whole field developed, to the skepticism, because the cells were very rare, there was lack of knowledge of the type of the cells, not very specific markers, that people started looking at other alternatives. And if you plot the cell-free NIPT, it's actually, it goes very well with this. So, people not having a lot of confidence in cell-based or, sourcing the fetal cells, then they start looking at the cell-free NIPT. Ironically, it was the same time when our company was founded. Not very intelligent decision, I guess, but it was. But the reason was that some of the founders of RCD had also worked in fetal cell field for many years, and they knew the challenges. So they took a more bottom-up approach. And what came out of that exercise was this list of a checklist. Saying that if a quality statement, if you like, so saying that if a technology claims that it can do cell-based NIPT, then it should follow these points. First was there should be knowledge of the type of the fetal cells that circulate in maternal blood. Connected to that was there should be antibodies that should be both sensitive and specific. So you need to enrich good number of fetal cells, and you should not classify maternal cells as fetal cells. The technology should be platform and parameter independent, and it's not only about counting the number of fetal cells. Once you have enriched the fetal cells, you should be able to have access to these fetal cells for performing downstream analysis, and there should be the DNA that you get from these fetal cells should be of high fidelity. So I'll basically walk you through this checklist. This is the paper that we published in 2013. So it was very interesting because we didn't have any knowledge of the type of the fetal cells, so we didn't know anything about the markers. But we use depletion or non-enrichment to get hold of 200 fetal cells. What we then did was we ran a gene expression profile on these fetal cells to see what are the genes that are being expressed in these fetal cells. And what we found was that the fetal cells that were enriched or were circulating in maternal blood were of extravillous trophoblast origin. And what are these extravillous trophoblasts? You have a diagram of the fetal-maternal interface. So you have the villi for the placenta that are anchoring the uterus, and so you have cytotrophoblasts here. Then you have a bunch of cytotrophoblasts which are part of the columns. And what happens is that by the end of the first trimester, some of these cytotrophoblasts, they shed from the column and they start invading the maternal decidua. And they are doing that because they are lining the arteries, the veins, and the glands for the vessel remodeling that happens in the first trimester. So once they shed from the column, now they are called extravillous trophoblasts. And they line these vessels. And we believe that some of these extravillous trophoblasts that are lining the vessels, they shed into the maternal circulation, and these are the ones that we catch by our method. And currently we are using, another interesting point is that cytotrophoblasts are epithelial cells. So when they are invading the maternal decidua, they change phenotype, they become mesenchymal. And currently we are having 8 markers that we use to enrich and identify these fetal cells. And some of the markers, they target the epithelial morphology or phenotype of the cells, and some of them, they are targeting the endothelial morphology of the cells. So we have this combination of markers that we're using. And last year, we secured a patent in the US for these markers that we use to enrich these fetal cells. What about the specificity and sensitivity of the markers? This is the data from 190 pregnancies. So out of 190 pregnancies, in the end of the first trimester, 99 were categorized as low risk group, and 91 were high risk group, based on the serum screen, the ultrasound, and the maternal age. And the data that we got out of that study was we were able to enrich 2,440 cells with a mean of 12.8, median of 10, and a range of 1 to 46. And here's the distribution. So the blue bars, so the number of samples plotted against the number of fetal cells, you have the blue bar for the low risk group and the red bar for the high risk group. We want to see if there's any difference in the distribution in the number of fetal cells based on whether pregnancy was high risk or low risk, but you can see that it follows very nicely. And then we were able to enrich fetal cells from every pregnancy, and that has been the bottom line of cell-based NIPD, that there were many no-calls before. What about the specificity of the fetal cells? So what we did, the idea was to see whether the cells that we call as fetal cells, are they true fetal cells, or they are maternal cells being classified as, misclassified as fetal. So what we did, we sourced 208 fetal cells from male pregnancies, and after they were identified based on the different classifications that we have to identify these fetal cells, we performed XY FISH, and this is the data that we got. So out of 208 fetal cells, 164 were confirmed as from the fetus. 32 cells didn't have any FISH signals. 12 cells were lost during FISH. But the encouraging part was that none of the cells that we had classified as fetal turned out to be maternal. And this is the part of the paper that we published in December last year. I briefly mentioned about the fetal cell morphology. So this is fetal cell among the maternal cells. This is the picture that you get after you scan the slide and then validate it. So we are using different classifications. We are taking into consideration the cytoplasmic staining pattern, the nuclear morphology, and we're also having a counter stain that goes on the maternal cell, not on the fetal, and also the size of the fetal cell. So these are some criteria that we have developed in-house to identify fetal cell among the maternal cells. Turnaround time, from the time when we have the sampling from the pregnant woman to the time when we get the DNA from whole genome amplification, it takes around 14 hours, roughly. So 1 hour for blood processing, 4 for enrichment of the fetal cells, another 3 hours to scan, identify, and pick the fetal cells. Then we collect them in PCR tubes and perform the whole genome amplification. But we are quite aware that this is not a high throughput method because it also puts a lot of pressure on especially the picking part is quite laborious and labor intensive. And we would like to make this procedure automated and also have fewer people or reduce human intervention. So what we are currently doing is that we are looking at 2 different platforms. One is the Multimax. So in the manual, you can only process 1 sample at a time. But then Milteney has another system called Multimax where you can have 24 samples that could be processed. So that also releases the FTEs. And we have tested this in our lab, and the number of fetal cells that we get out of this versus the manual is the same. Another thing that we are currently pursuing, we have just initiated a project in collaboration with Aarhus University in Denmark, where we would like to go away from the manual validation of fetal cells, because the fetal cells are fluorescently labeled, and we could actually, in principle, use FACS instead of the scanning of fluorescence microscope. But we have initiated, we don't have any data currently. But if we automate this, we'll of course make it quicker, but also release the FTEs and also the fact that we won't have any human intervention. Some other things that we have tested in the lab, which we think is quite important for the clinical utility of a cell-based NIPD. We have tested different blood collection tubes. We have waited before the blood processing up to 72 hours, and we have also transported the blood and processed after 72 hours without any difference in the number of fetal cells that are retrieved. And as I also mentioned before, we get fetal cells from every sample. The last point in the checklist is that the DNA that comes out of these fetal cells should be of high quality and quantity. So this is just a brief introduction to what we are doing currently. We perform whole genome amplification. So we have standardized it. We have tested one cell up to 7 cells, and we use the picoplex kit. Then we perform array CGH using the Agilent kit. So I'll just quickly go through some of the cases. This was one of the 2 cases that we published in collaboration with Baylor College of Medicine in December last year. And this was the first one where we performed array CGH on the cells that we had picked using our method. And so it was a high-risk pregnancy that was offered amniocentesis. The blood sample was collected before the invasive procedure. The 2 fetal cells were enriched using RCD method, and whole genome amplification and array CGH was performed in Baylor College of Medicine. Because it was a mosaic case of monosomy X and ring X, so some of the cells had complete loss of chromosome X, and some of the cells had distal or partial loss of chromosome X. So we decided to pick these cells individually. So what you see is cell number 1, and that's the genotype you get. So there's a complete loss of chromosome X, and the cell 2, 450, have a partial deletion on the distal arm of chromosome X. And this is the result from the amniocentesis. And then we also performed NGS with the same result. So you have the complete loss and you have the partial loss of chromosome X. Going further, this is a paper that was accepted just last week to be published in Prenatal Diagnosis, where we have showcased 5 different cases. So it's basically, can we look for CNVs? Can we go beyond common aneuploidies? And this is the table from that paper. So you have the first case with trisomy 21 on 7 cells. Then you have the case 2, trisomy 13. Case 3 was trisomy 2. And I would like you to focus on the last 2 cases. And this was on 5 fetal cells. And there's a partial duplication on chromosome 21, a 12.4 megabase duplication. And the last one is a structural rearrangement between chromosomes 4 and 8. So 31 megabase deletion on chromosome 4 and 30 megabase duplication on chromosome 8. And this is the array CGH results that we got. So on top you have the partial duplication of chromosome 21. And then you have case 5, where you have the deletion in chromosome 4 and duplication on chromosome 8. And this is from the male pregnancy. So what we do, we normally have the control DNA from the opposite gender. So you have the loss of the X and the gain of the Y. And this is just the opposite, the gain of the X and loss of the Y. And then we have the CVS results from the same case. So you also see it's very nicely represents here the partial duplication and the deletion and duplication on chromosomes 4 and 8. And this is a case that we just got it last week in our lab. This is a 4.9 megabase deletion on chromosome 3. Here you go. And then we got the CVS results, and you can also see it there. What we are currently doing is, so these are, of course, would appreciate that these are not, consecutive samples. We are, choosing and picking and choosing and showing you the data. But what we would like to do now, we have locked the protocol in terms of the fetal cell selection, picking, whole genome amplification, array CGH, and initiated a clinical validation study in collaboration with the clinical genetics department at the hospital. And this is an ongoing study. So what is going to happen is that we have collaborated with 6 different hospitals in Denmark. We are recruiting high-risk pregnancies who are undergoing CVS. The inclusion criteria is that these pregnancies should be singleton pregnancies between the gestational ages of 10 to 13, and which are opting a CVS. So the idea is to collect blood for cell-free NIPT, then we also collect blood for our cell-based NIPT, then we also correlate the data with the invasive method. And there are 11 cases up until now that I would like to show. And we're really lucky that we have a trisomy 21 and 13 among these 11. But if you look at 2021, it was not called by cell-free NIPT because of the low fetal fraction, and then it was performed again, and then it was called. And this was an interesting case. So the woman went for ultrasound. She agreed to go for cell-based NIPT, give blood for our test. She came back after a couple of days and then realized that she has a spontaneous abortion. So that's why we don't have any data from the CVS. But we know that trisomy 16 is a very common cause of spontaneous abortion. And then we have a trisomy 13, which was called by cell-free NIPT and by cell-based and CVS. Going further, we are looking at launching a cell-based NIPT test in Denmark by the end of this year. So what's going to happen is that 1/3 of the pregnancies that are classified as high risk, they opt for non-invasive NIPT. So they are just given a cell-free test. But what will happen is that we will also sample them for cell-based NIPT, and then whatever will come positive will then be referred to for CVS. In brief, we have identified the type of the fetal cells that circulate. We have tested markers which are both sensitive and specific to enrich these cells. We have a robust method of enriching fetal cells. We have picked the cells for downstream analysis, and apart from finding common aneuploidies, we have also looked for CNVs. And as I also mentioned before, we have very close collaboration with Aarhus University Hospital, Department of Gynecology and Obstetrics, and also Department of Clinical Genetics. And this is a team that we have at RCD Biotech. Thank you so much.